Hello, besties. So today we're reading a scientific article. Wait, stay and watch the whole video. Okay, so also a bit news about the channel. I might start posting videos on Wednesday, or I might keep posting videos on Thursday. I am not sure. See next week. If I don't post a video by Wednesday next week, the video is gonna come out on Thursday next week. Okay? So anyways, onto the video where today we're going to be reading a scientific article. Before you ask, yes, it's 21 pages. How are we going to read this? Well, I basically looked up an article and basically said how do you read scientific papers, and they asked a bunch of old scientists. I don't know if I should be doing that. That might be cause flashing lights. So they said the first step is to read the abstract. Okay, so let's do it. Abstract. Oh, wait. Marine Sponges as Pharmacy by... I tried looking up some of their names. It's by Detmer Sipkema, Maurice C.R. Franzen, Ronald Osinga, Johannes Tramper, and Renee H. Wiffles, Vichels, or Wichels. Any one of those. That's all the types I saw. They're from the Netherlands, so I think it's like Vichels. Renee Vichels. I hope that's correct. Sorry if it isn't. So abstract. Said first read the abstract. Marine sponges have been considered as a gold mine during the past 50 years, with respect to the diversity of their secondary metabolites. I don't know what that word is, so we'll look it up later. The biological effects of new metabolites from sponges have been reported in hundreds of scientific papers, and they are reviewed here. Sponges... Oh, I'll stop doing... No, I'm gonna keep... Reviewed here. I'm not gonna do the news voice anymore. Sponges have the potential to provide future drugs against important diseases such as cancer, a range of viral diseases, malaria, and inflammation. I had malaria once when I was a baby. It was not good. Although the molecular mode... Malaria and inflammations. Although the molecular mode of action of most metabolites is still unclear, for a substantial number of compounds, the mechanisms by which they interfere with the pathogens of a wide range of diseases have been reported. This knowledge is one of the key factors necessary to transform bioactive compounds into medicines. Sponges produce a plethora of chemical compounds with widely varying carbon skeletons, which have been found to interfere with pathogenesis at many different points. The fact that a particular disease can be fought at different points increases the chance of developing selective drugs for selective targets. Keywords: sponge medicine, space natural products, space cancer, space inflammation, space virus. Okay, let's look up what metabolites mean. Found it. And now let's look up something else. Pathogenesis is the progression of processes of cellular lineage, maturation, becoming mature, and migration, and eventual morphogenesis of both individual cells and their architecture and forming tissue and organ. Let's look. But this, Wikipedia says, is the process of which a disease or disorder develops. Okay. And also progression and maintenance. So basically, biological. So metabolites, chemicals that cause metabolization. Basically, have you heard about metabolism? It's like that. The chemicals that cause metabolism. You can look up, if you don't know what metabolism is, you look up metabolism. I believe in you. So, metabolism. Chemicals that cause metabolism. Well, they're secondary secondary chemicals that cause metabolism. Biological. And it's like, they're not sure, like, although the molecular mode of action of most metabolites is still unclear, for a substantial number of compounds, the mechanisms by which they interfere with pathogenesis is been reported. So they know how these chemicals interfere with the creation of diseases and stuff like that. Mode of action, like how they do it. Look up mode of action for me. Anyways, so now we just skip the rest of this and go to the conclusion. What did they find out? Wait, no, we're supposed to look at data first. Oh my gosh, so many graphs. Okay, don't be scared. Homeopathic drugs. Is this figure? This is figure 2. And what's figure? Oh, that's figure 1. Figure 2. No, anti-inflammatory, anti-tumor, immunosuppressive, or neurosuppressive, antiviral, blah blah blah, just basically anti-diseases. So I'm guessing these are like chemicals. This is how the chemicals look. Some of these sponge-derived molecules. Anyways, let's keep going. Why is it sideways? Anti-inflammatory products. These are examples of that. Is it just talking about like the history of this? Interesting. Inflammatory cascade inside the cell. I think these are amino acids and these are polypeptide chain. Basic amino acids are protein. Well, not our protein. Amino acids make up, I think it goes, amino acids make up polypeptide chains. Basically chains of amino acids. And then they fold into shapes. And those shapes are the proteins. Wait a minute. But that looks like fat, though. With a tail. I'm confused. Oh, wait, it's not amino acid. It's arachidonic acid. Wait, I need to look up something. That is not a polypeptide chain. Sorry. This is lipids. Basically, at least this looks to me like fat. Basically, this is fat. Yeah. Okay. So, I think this is the data now. Sorry you have to tilt your head. This is so weird. Why is it Wait, can I do something? Wait. Bestie, I'm so smart. Okay. Compound. Okay, let's just skip to the conclusion. Well, no, that's the sources. Okay. Okay, conclusion. Marine sponges produce an enormous array of anti-tumor, antiviral, anti-inflammatory, immunosuppressive, antibiotic, and other bioactive molecules that can affect the pathogenesis of many human diseases. The relationship between the chemical structures of the secondary metabolites from sponges and the diseases they affect is usually not obvious. Different components affect the targeted disease by different mechanisms. For example, microtubule stabilization or interaction with DNA to combat tumors. Moreover, inhibitors of transcription may be effective against both cancer and viral diseases. To make things more complex, there are many relations between. For instance, inflammatory, cancer, and viral infections via the immune system, which plays a key role in certain responses of the body to these diseases. Chronic inflammation of the lungs by cigarette smoke often leads to lung cancer and cervical. Wait. Oh, I was just kind of reading that. I wasn't exactly absorbing it. How did we. Wait. To make things more complex, there are many relations between, for instance, inflammation, cancer, and viral infections via the immune system, which plays a key role in certain responses of the body to these diseases. Chronic inflammation of the lungs by cigarette smoke often leads to lung cancer, and cervical and liver cancer can follow chronic inflammation caused by papillomaviruses and hepatitis B and C viruses, respectively. In addition, I feel like I read this before. In addition, limited activity testing only on cell growth inhibition and not on antiviral properties yields an incomplete overview of the actual properties of the metabolites. Finally, for many bioactive molecules for sponges, the exact mode of action and their origin, sponge or symbiont, are still unclear. Most bioactive metabolites from sponges are inhibitors of certain enzymes, which often mediate or produce mediators of intracellular or intercellular messengers involved in the pathogenesis of a disease. As this is usually a cascade of reactions inside the cell or tissue, many enzymes in the cascade are targets for potential therapy. The different enzymes in case in the cascade. Okay, wait, my 15-minute timer is almost up, so I'll finish the conclusion. Right? Now, okay, where was I before I started speeding up? In addition, limited activity testing, only on cell growth inhibition, and not on antiviral properties. In addition, limited activity testing yields an incomplete overview of the actual properties of metabolites. Finally, for many bioactive molecules from sponges, the exact mode of action and their origin, sponge or symbiote. Maybe symbiote just means like fake sponges, are still unclear. Many bioactive metabolites from sponges are inhibitors of certain enzymes, which often mediate or produce mediators of intracellular or intercellular messengers involved in the pathogenesis of a disease. Okay, so like intracellular means like inside. I think it means within the cell. And intercellular means like to different cells, like intergalactics. Like something that says intergalactic would be like between galaxies, like, oh, let's send this intergalactic message to Andromeda. And like Andromeda could send it somewhere else. I don't know. There isn't anything at Andromeda to send things to. But like you get what I mean. Like inter is like between things. Intra is like inside. Like intragalactic travel. I can travel inside the galaxy. Okay. Blah blah blah. As messengers involved in the pathogenesis of a disease, and this is usually a cascade of reactions inside the cell or tissue. Many enzymes in the cascade are targets for potential therapy. So they want to stop these enzymes that start the birth of a disease with the chemicals the secondary chemicals that do metabolism in sponges. So they want to use those chemicals to stop the enzymes that can cause the birth of a disease. Am I getting that correctly? To potential therapy. The different enzymes in the cascade can be structurally completely different proteins. Therefore, it is not surprising that a wide range of metabolites could be used for the treatment of one disease. This applies in particular to a complex disease, such as cancer, which is affected by so many different factors. Furthermore, antiviral molecules also appear to encampse a wide array of chemical structures, such as peptides, lipids, alkaloids, sterols, such as peptides, lipids, alkaloids, sterols, and oligonucleotides, and phenolic macrolide. Okay, no, let me say this word. Oligonucleotic, nucleo, okay, what is it, oligon, subscribe, no, I can't, what, did I just say subscribe? we are not gonna dwell on that. Okay. Oligo, oligo, allele, nucleotides, oligo, oligonucleotides, and phenolic macrolide. All of these are chemicals. A similar diverse pattern is observed for antibacterial and immunosuppressive metabolites. Most compounds that display anti-inflammatory activity are sus. They're sus, guys. Are sesterterpenoids. Cisterterpenoids. Neither the compounds that display. Nevertheless, in these cases, the activity of sponge metabolites is concentrated on certain steps. For instance, most inflammatory compounds act against phospholase A subscript 2. Basically, they're just sending these metabolites against from sponges, remember, like the animal, the type of animal. If you didn't know, sponges are animals. So basically, they're using these chemicals that are used in metabolism, and they're basically throwing it at certain, like, steps in the process of a virus being born. Not a virus being born, silly me, a disease being born. Anyways, the potency of sponge-derived medicines lies in the fact that each of these thousands of metabolites and their derivatives has its own specific dose-related inhibitory effect, efficiency, and potential diminished side effects that determine its suitability for medicinal use. In addition, the skeleton or active core of these molecules may be used as a vehicle to develop derivatives with their own specific efficiency and side effects. Therefore, the most important challenge in transforming bioactive molecules into medicines is now to screen the treasure house of sponge metabolites and select those that display a specific mode of action with the desired characteristics against the disease. An important question for the future remains how to actually prepare the potential novel drugs on a larger scale. So what was the point? I don't know if this was like we did an experiment, or was it like let's just talk about it? You know what? If they really was just like let's just talk about it, honestly, props to them. Let's just talk about this interesting thing. That's cool. Should we read the introduction? It's like this. All the way to like that and that, and then that. Should we? This is, or is it just like let's look at the introduction. Let's just the relationship between sponges and medicines goes back to Alexandrian physicians and was thoroughly described by the Roman historian Plinius. Do you know Plinius? We know Plinius. Physicians used sponges that were saturated with iodine to stimulate coagulation of the blood, or with bioactive plant extracts to anesthesize patients. Sponges were soaked with pure... Wait. Yeah. Sponges were soaked with pure wine and put on the left part of the chest in case of heartaches, and soaked in urine, to treat bites of poisonous animals. Plinius, yes, the same one from before, recommended the use of sponges against sunstrokes, and they were used against all kinds of wounds. Bone fractures, dropsy. What's dropsy? I don't want to look it up. Stomach aches, infectious diseases, and testicular tumors. Oh. Oh, ancient Romans. Oh, there's more. Or even as implants after breast operations. At least since the 18th century, Russian, Ukrainian, and Polish physicians have been using a freshwater sponge they call badiaga. Badiaga? Badiaga. I said that wrong, sorry, for the Russians, Ukrainians, and the Polish, for the treatment of patients. The dry powder of this sponge is rubbed on the chest or back of patients with lung diseases, or the sore place in case of a foot or leg aches, such as rheumatism, Ofijalski discovered that Badiga, I think that would, I meant to say Badiaga, that Badiaga is not really one sponge, but mixtures of several freshwater sponges. Wait. I have to go to overdrive. Freshwater sponges that differ depending on the region. In Poland, it consisted of powder of Euspongilla lacustris, Ephidatia fluviatilis, and Meania Moyeri. Well, the Russian Badiaga was a mixture of Espongilla lacustris, Ephidatia fluviatilis, and Spongilla fragilis, and Carterius Stepanowi. He suggested that high iodine concentration in all sponge species give rise to the wholesome effect of Badiaga. As presented, Stodal, syrup containing roasted Spongia officinalis, is used for homeopathic treatment of dry and asthmatic cough in the Western world. And you know what? We're gonna stop reading that introduction there. It's called, this paper is called Marine Sponges as Pharmacy by Marine Biotechnology, by Detmer Sipkema, Maurice Franzen, Ronald Osinga, Johannes Tramper, and Renee H. Wilhus. I hope you enjoyed the video. Like and subscribe. Comment down below what was your favorite part. And peace. Drink water and eat your food.